So our topic is mechanism of acid-fast staining. We are going to talk about why some bacteria acid-fast, why some bacteria non-acid-fast. So acid-fastness is due to the lipid-rich mycobacterial cell wall. Right? The lipid is mycolic acid, which is found in the mycobacterial cell wall. Right? So what happens is lipid, when there is a lot of lipid in the cell wall, it resists decolorization. Right? Decolorization is the second step of acid-fast staining. So when you add the decolorizer, the acid-fast bacteria have a lot of lipid in their cell wall. It prevents the entry of the decolorizer into the bacterial cytoplasm. So we'll talk about in detail. Right? Acid-fast stains involve three sequential steps. Number one is first step is carbonfuchsin, which is the primary stain. You apply it for about 5 minutes. Right? So the lipid solubility of carbonfuchsin allows it to penetrate the lipid-rich mycobacterial cell wall. Right? There are two modifications of acid-fast stain: Seale-Nielsen modification and the Quinone modification. In the Seale-Nielsen method, we use heat to enhance the penetration of carbonfuchsin into the bacterial cell. We use heat, that's why we call it hot carbonfuchsin. We use hot carbonfuchsin. But in Quinone modification, we use a chemical agent called tergitol, which reduces the surface tension, so that it also enhances the penetration of the dye into the bacterial cell wall. Right? That is Quinone modification. Basically, Quinone modification uses carbonfuchsin, but it doesn't use hot carbonfuchsin. It uses the wetting agent to enhance the penetration of the carbonfuchsin. Then the second step is acid alcohol, which is 3% hydrochloric acid, 95% alcohol. Sometimes there is an alternative decolorizer, we call it 20% sulfuric acid. The counter stain is methylene blue or malachite green, which we apply for about 2 minutes. Right? So this is the important step. We are going to, our topic is mechanism. So now we are going to try to understand the mechanism. Why some bacteria become acid fast, why non-acid fast. Right? So how? Right? So we are going to see how it works. So first step is, you can see all the bacteria are colorless, right? Before staining, right? Then we add the carbonfuchsin. It will stain all the bacteria, both acid fast and non-acid fast bacteria will appear red. The second, right? You can see carbonfuchsin. They both types of bacteria are red. So when the decolorizer is added, it will decolorize all the bacteria except acid-fast bacteria, because the acid-fast bacteria are resistant to the entry of decolorizer. So the acid-fast bacteria will not be decolorized. Non-acid-fast bacteria will be decolorized. Right? That's what you see in the third picture. Right? So the acid-fast bacteria remain red. Non-acid-fast are decolorized. They are colorless. You can't see them if you look to the microscope. Right? Then you have the counter stain. Counter stain, we colorize only the decolorized bacteria. Right? Acid-fast bacteria, they are already red. They can't be colorized again. Right? So the non-acid-fast bacteria are decolorized, so they will be colorized by the counter stain. If you use methylene blue, they will be blue color. If you use malachite green, they will be green color. Right? That's what you see. You see with the counter stain, acid-fast bacteria remain red. Non-acid-fast bacteria, here we have used malachite green, so they became green. So this is the mechanism of the acid-fast staining. Hope you enjoyed the lecture. Thank you. If you found this video helpful, please like this video and subscribe to my channel. You can get new video notifications by clicking the bell icon. My Patreon supporters can request lectures and get exclusive content. Please check the description for links and more details. Thank you.